You can start now, sir.
Oh, thank you. Thank you so much. Good evening, my dear students. You are all are warm. Welcome to the wonderful session of the biology class. Once again.
Good evening, sir.
Good evening. Today, what we are going to discuss about, the topic side heading is known as the AIDS. Already know about what is meant by AIDS, isn't it? AIDS. What is the expansion of AIDS? It's acute immunodeficiency syndrome, isn't it? Sometimes they were asked the question: the expansion of the AIDS. What is it by immunodeficiency? Will you please? What is by immunodeficiency? What is by syndrome? That thing is must note it down. Sometimes they were asked questions on that. Yeah.
So syndrome is when the symptoms, sir, it is like based on it appears in a large number of people and all of them show like similar symptoms. So it's together they call it syndrome, sir.
Yes. Okay. Correct. Exactly correct. Okay. The word AIDS stands for acute immunodeficiency syndrome. Already you know what about. It means deficiency of immune system occurred during the lifetime of an individual, indicating that it's not a congenital disease. Already you know very well what is by congenital disease, isn't it? Okay. It will not start from the in. Okay. It will receive from the life period, in between the period, due to some unwanted things. Okay. See, AIDS, acute immunodeficiency syndrome. This is caused by the causative organism is HIV. The HIV is also stand for human immunodeficiency virus. Got it? Okay. See, what is by syndrome? Already, Ajay started the statement, or Ajay also gave an explanation. Syndrome is a group of symptoms. AIDS was the first reported in the year 1981 in America. And in the last 35 years, so it has spread all over the world, killing more than 25 million people. This is the state. In India, 1984 have been reported. Okay. So this is the statistical even. Now you must note it on 2022 onward. What are the cases analyzed? Statistical things also you must note it. Okay. Then we'll pass on to the next one. See here. This is the human immunovirus deficiency virus structure. Already know very about. Already know very about the HIV, isn't it? It has a two components. What are the two components? Is very, very essential. The outermost covering is made up of glycoproteins. They may be depends upon the nucleotide and also that amino acid sequence. The glycoprotein is here. See that a glycoprotein 120, and another glycoprotein is number 41. What is the difference between these two? 120 and 41 depends upon their amino acid sequence. The structure will be different. Inside there is see that structure. There is a bead-like structure is there that is called as matrix protein. That is P74. And at internal there is a covering of nucleic acid that is called as nucleocapsid protein, the P24. Inside see the structure, the DNA molecule is here. This is a single standard RNA molecule. two copies. Not only that, and also the presence of the one of the enzyme is called integrase. Not only integrase, it is also inside there is one of the protein molecules. Very, very important thing. Super transcriptase enzymes. So collectively called as human immunovirus. Not only that, there are so many protein molecules also there, and also proteases enzymes also there. The cell wall is also made up of which one lipid bilayer membrane. Okay, so these are all are construct the viruses, particularly single standard RNA retroviruses. Okay, so this is the structure. We will see clearly one by one once again. Okay, see the same structure is here. Okay, so this is called as what is that outermost? What is that? The shield. There are so many protein molecules. The upper part is called as 120 glycoprotein. Lower part is called as glycoprotein 41. Inner side, what are the protein molecules? Also, the proteases enzymes, matrix, nucleic acids covered the internal molecules. Why it is called as nucleic acid? No, the protein layer covered the internal molecules, particularly nucleic acid. With the of the two enzymes called one is called integrase, another one is called as reverse transcriptase. Okay, go to the next one. See here. So virus or pathogen. Here, who discovered the HIV virus? It is one of the pathogen. They only produce the disease one. The retrovirus HIV. You note it down. This is very, very important. Already I told you. This is the structure. Retrovirus always look like a round shape. Okay, and also inner side there are so many molecules also possesses. See here, the scientists who discovered the virus, because AIDS, Dr. Luke Manteginier, human immunovirus deficiency virus, and also called as LAB. Human immunovirus, otherwise called as HIV virus, also called lymphoadenopathy associated virus, or HTLV3, human T-cell lymphotropic or leukemia viruses. These viruses, a group of called, what is that? Retrovirus. Once again, these are all, what is that? Related to the, similar to the HIV virus, the structure and also the properties also. Okay, so see that this virus belongs to a group called as retrovirus. Okay, the structure, what will happen? See that outermost. See, we are seeing the Dr. Luke Manteginier, who discovered the HIV virus in America.
Sir?
Okay, yeah.
So why is it called retrovirus, sir?
Because its structure will be similar. Structure will be different shape compared to the other one. Okay, each and every retrovirus, they are having more specific pattern of structure. That means the outermost covering look like which one the protein molecule. Okay, inner side there is a membrane is there, and inside there is a nucleo capsid also there. Then inside is a DNA molecule. Such type of structure only present in the retrovirus only. Okay, this type of, that means outside to inside when you are moving, so there is a needle-like structure. It is made up of two proteins, and also inner side there is a membrane is there. What is this? This membrane is called as, what is it? See here. So bilayer is also covered in the outermost. Inner there is another layer, and again it's also another layer. Okay, inside there is a nucleic acid. Likewise, such type of viruses called as, what is that? Retrovirus. Again, take an example. Bacteriophages. Only hexagonal shape only it, isn't it? Inside there is nucleus only. Take an example of tobacco mosaic virus. What is that? The capsid is the outermost layer covered. Inside, what is it? There is a single standard DNA molecule. Only that there is a component present. But in this category, there are stages. There are so many layers. That outermost layer, the middle layer, and also innermost layer, and likewise, that's the reason. They are all called such type of viruses called as What is that? Retrovirus. Similar to that virus, so many viruses also present in it. What are they? LAV, HTLV3, and leukemia viruses. So all are having the similar virus, similar structures. They are all are considered getting known as retroviruses. Are you clear now? Very clear, Ajay?
Yes, sir.
Okay. Okay. So you are seeing the Dr. Nick mentioned here in the 81. So 1981, it is called the HIV. Okay. How it will be? See here, which has an envelope consists of lipid layer. Lipid is the outermost layer, envelope. And bilayer derived from the host cells. This is also derived from the host cells. And the membrane and projecting knob like glycoprotein spikes that already told the spike like structure is outermost. What is that? Spike. Similar to that of the which one COVID. COVID also it has the which one spike like structure. That is the modification. There are so many modifications occurs. Okay. With a pedicel formed of virus coded glycoprotein. What is that? See the structures. See the structures. This is the structure of glycoprotein, isn't it? See here. So glycoprotein. Okay. Covered by a bilipid layer. So that is the reason. This protein layer is also completely covered the bilipid also. So that is the structure of viruses. See here. And this envelope encloses the RNA genome. Just note it down. This is very, very important. A single standard RNA filament is segmented into two identical filaments. Both are similar in nature. That's why they have given us the single standard. Once again, repeat for me. Single standard is denoted as small s and small s. Okay. Double standard means DS. C denoted as circular DNA. Okay. These are the symbol you must note it down. Mostly in the HIV category or retroviruses. Which one is the nucleic acid? Single standard RNA molecule. How many copies present in it? 2 copies with associated with the reverse transcriptase enzymes. This is the important point. Okay. How many copies of reverse transcriptase? It is also 2 copies. DNA common is the 2 copies of DNA molecule. Sorry, RNA molecule. Extremely sorry. RNA molecule. Single standard RNA molecule. Then single standard RNA molecule, how it will produce the DNA molecule with the help of the enzyme is called as reverse transcriptase. What is meant by transcription? What is meant by transcription? Anybody else? Anybody else, my dear? See here. So this is a structure. See how many protein molecules? These are all are simply called as protein. Which one is a GP denoted as glycoprotein? Okay, because enzyme is there. That is called as integrase is also one of the protein molecules. Okay, already in our report, all the protein molecules are not an enzyme. All the enzymes are protein. Okay, you got it. So single standard RNA molecule. See, this is the single standard. How many copies is there? 2 copies. This is called as what is then P10 proteases in. This one is called as reverse transcriptase. Reverse transcriptase. The protein molecule 64. It may be denoted as the amino acid sequence, I think. Okay, see here the 2 transcriptase. Enzyme is also there. Listen. See, next, the HIV, the scientist who discovered the virus that caused AIDS has died. Luke mentioned it. It consists of a core DNA with a reverse transcriptase surrounded by a protein coat. Okay. The core also has 2 types of protein molecules. That is, the inner protein coat and also the outer protein coat. The inner protein coat is the protein molecule P24, and the outer protein coat is the P17. These are the core. Okay, see that. Go to the next one. Is the core is covered by the phospholipid bilayer, studded with the glycoprotein. That glycoprotein is 120, and also glycoprotein 41. Sometimes they may ask the question from this area. How The glycoprotein of HIV molecules may be interchanged like this. 120 means they may give sometimes 210. Here 14. You may confuse that time. So you have to clear in this area. Okay. So glycoprotein 120 and also 41, which is GP 120. And diagrammatical question, they may ask the first question. That time you have to clear. Okay. See here. The glycoprotein 120 has a complementary sequences to CD4. CD4 receptor. What is the meaning of CD4 receptor? Already present the T cell. CD4 receptor. Receptor is also one of the protein molecules. It receives the, which you know, the protein molecules from the otherwise called antigen secreted by the HIV. The receptor present on the macrophages. Macrophages. Okay. The macrophages, the cell wall or cell membrane containing the CD4, and also it is also made up of glycoprotein. It can be easily attacked with the which one you know T cell. Okay. As the result, what happened? The virus easily attacks the which one you know T cell receptor. The T cell receptor is called as CD4. The CD4. Got it? Okay. See, in retrovirus, the enzyme reverse transcriptase is used to synthesize DNA from RNA. That's why I asked you, what is like transcription? Transcription is nothing but the DNA to RNA. But in this category, reverse transcriptase, okay, the HIV contains RNA molecule. Okay, single stranded RNA molecule. The single stranded RNA molecule produces the double stranded DNA molecule with the presence of the reverse transcriptase enzyme. That's why the process is called as what is that? Reverse transcription. Once HIV produces the DNA from its where RNA, the DNA is integrated into the host cell DNA. Once what happened, you know, first you should know here, RNA is the genetic material of HIV. It produces the DNA molecule. This DNA only incorporate with the which one you know host DNA. As a result, what happened? You know, the DNA molecule mingle with the host DNA. It produces the viral protein molecule. It comes under what happened? It control all the activities of the host cell DNA. So as a result, what happened? You know, it also incorporated both the DNA molecule. Otherwise, both the genetic materials are united together. So what will happen? You know, it will produce the which one? The host DNA also mingle with the virus DNA. The viral DNA completely control the host DNA. Okay. So as a result, what happened? It produces a new gene, means it is also which one you know virus oriented only. So that is the process. See, that it can remain dormant, giving a sigh of it is present, it can be over the coast. Cell genetic machinery to produce more viruses. Suppose all the coast controlled by which one you know the viral DNA, means what is that? It can produce their own what is that genetic material, and also they produce own what is that viruses. Virus, that means genetic machinery. It produces number of viruses because it all the control done by which one you know HIV. Okay, the production is also similar to that of which one you know viral dependent DNA, like that. The major cells infected B HIV is the helper T lymphocytes that bear the CD4 receptor. T lymphocytes is very important for the fighting of many diseases. They are having the more important and most important of the immunity power. Okay, when it bind with the which you know the HIV, okay, bind with the T lymphocytes, bears the T4 receptor. The receptor without receptor, it is not bind with the HIV molecules. Okay, so that is the question. HIV molecules means what is that? Antigen produced by the HIV viruses. The attachment of virus to the CD4 receptor site is by the help of glycoprotein 120. Suppose the research also going on. Suppose the glycoprotein 120 is not present in the virus, what happened? The CD4 receptor site completely lacks, so it cannot bind with the which one you know receptor site. As a result, the person is curable, isn't it? That thing's also going on in research oriented. And the protein coat of the viruses. This is very, very important. How it will be attached to the T lymphocytes with the presence of CD4 receptor. This is very, very important. CD4 receptor is help to bind the which one you know? Glycoprotein 120. Okay. So this is the see here. So this is called as a CD4 receptor. See here, this is a CD4 receptor present in the cell membrane. Here there is a glycoprotein molecule that is called as Hooks. It is easily bind with the CD4 receptor. Then only it may inject the molecule, which one is the DNA molecule, also insect. As a result, what happens? All the control under HIV viral genetic. So as a result, they produce more number of virus-related molecules. Finally, what happened? It's not produced. So lymphocytes completely denaturation. Okay. So this is the virus HIV. See here, there is a HIV is also bind with the receptor cell, and also it includes the genetic material. Okay. See here, this is the steps. These are all a very complicated one. So just to go through it, it will be better for you. Nobody, they will not ask in this area, inside what will be the mechanism of it. They may ask the question from this one, HIV, where it attached to on it in the T lymphocyte, like that. CD4 receptor, it's also made up of protein molecules because the presence of glycoprotein 120. Note it down. It is not in the glycoprotein 41. Okay, see that. So these are all on the another part. Okay, see here. Okay, see, these are all remarks related to that one. Okay, next we will pass on to the next one is mode of transmission. How the HIV is transmitted from one person to another person. Here, main category they have been given in the 4-part. The first one is called a sexual contact, and the second one is called parental. Okay, and also milk during the milk, mother milks to the fetus also, and then blood transmit an affected person to the normal person. When the blood transfusion takes place due to some accident, unexpectedly they will introduce. As a result of what happened, infected virus also enter. And then a third one is the last one is called as non-sterile tools. Just when you are going to the hospital, okay, in ancient ages, a single syringe used for many people. Okay, but nowadays they use disposable syringes there. Okay, in previous category, what they were used, only one syringe is there, but one needle is there. They are used for 20 to 50 persons. Okay, after that, come to the normal. So these are the, what is that? Transmissions. Okay, HIV transmissions done by these are the things. So next one is, what is that? Majority of the respondents agreed that HIV could be transmitted through a sexual means, sexual contact only. Okay, 96.2%, and blood transfusion 96.7%, and sharing of sharp object, that is 92.5%. Maximum that category would be these two only, because a few of the respondents believed that HIV can be transmitted through the sharing of drinking cups also. Okay, 9.4%, and also mosquito bites. It is very complicated. I am not sure, but in this category, because suppose mosquito bites in the affected person to another person, it will be nowadays what happened. Completely there are human beings as well as the living organs completely nil in nature. But in this category, they told mosquito bite is also spread, which is 13% of 13.6% of HIV. But in this category, I have a doubt. Okay, just refer it and you will get it back. Next. Sexual contact is the most common mode of transmission, already in our tactics also. Okay, this is the first one. And second one is transfusion of contaminated blood, and also blood product. Blood product is nothing but in our serum or plasma, already in our, which is also one of the liquid part of the blood, isn't it? Okay, it can be, it contain what is that? Pathogen, otherwise called as the viruses, easily transfer from the one person to another person due to what is that? Blood transfusion, Sometimes there is a wound is there. Okay. When you are touching the affected person blood, what happens? Automatically we penetrate it in the in the way also. Okay. See here, and syringe and blade, plasma and blood. Already we discussed these two things. The first one is transmission, second one is transfusion, and third one is by mother to child, by the placenta, by the mother milk. It can be transferred another method. Suppose when the mother is also affected by the HIV, it can be easily transferred through placenta, otherwise called as umbilical cord, the connection between the fetus as well as the mother. So these are the three major category of the transfer of HIV from the affected person to normal persons. Suppose any one of the blade, okay, we were used syringe also. These are the indirect methods. This is another. See that the most common mode of transmission of HIV, already told it, the sexual transmission and parental contaminated drug injection, incipient, and also paraphernalia. Another one. Transfusion blood and the blood product. The blood product means what is that? Serum or plasma. Suppose take an example, HIV persons, okay, completely remove the plasma. One of the techniques also they were used to completely remove of the plasma. Okay, they were used the new one. What will happen? It will not beneficial because already a small part of virus person can be easily multiplied. Within 1 hour, how many viruses can multiply? Millions and millions. Okay, that is a question. And perinatal vertical transmission also takes place. And transmission from mother to child. How? Already you know. Through way of the umbilical cord, also it can be easily transferred. May occur during pregnancy, delivery, and also or the postpartum and fast feeding, milk, breastfeeding, like that it comes due to the through the milk. Say that sex, drugs addicts, blood transfusion, pregnancy, non-sterile. These are the HIV/AIDS is transmitted. It is not transmitted. How? When you are touching your friend's hand, shaking hand through the food, and also something else. Okay, insect bites. Here the insect bite is not spread. There, what they have given, 13% it will be possible, like that they told. In the pool, when you are swimming, bathing together, it will not spread. So investigation is very, very important. Okay, investigation first. There are so many types have been reported, but you must note that there are two types of is very, very important. One is called a screening test. Screening test. Once again, screening test means what is that? Whether the person suffering from AIDS or not, like that. ELISA. Okay. Enzyme linked immunosorbent assay. Already I asked the question, what is by sorbent? Okay. So enzyme linked immunosorbent assay. It is simply denoted as ELISA. And this is the screening test for the HIV. This is also sometimes they were asked the question. And a confirmatory test for AIDS. Confirmatory means the person got fear, 100% surely he got HIV. That test is called a Western blot method. Note it down. This is very, very important. Confirmatory test, Western blot. Screening test, ELISA. Okay. Detect the antibodies and proteins in patient serum. That's why we are taking the serum for the antigen antibody reaction. There is ELISA test is also another antigen antibody reactions also noted that, and also enzyme and the substrate complex also that. When you are studying the enzymology, we'll discuss very different. Okay. Sure. And then third one is antibody test, antigen antibody test, and also another one is also NAT test. Okay, these are the test for personalized. But here, these are the two is very, very important. Not only that, in this category, the direct test is also there, indirect test is also there, one step test and two step test also like that. And oral, nowadays there are the oral quick test is also present. In the oral quick test is what is the meaning of then the person suffering from the AIDS, we may collect the saliva from that person, we can analyze the HIV whether present or not. Okay, these are the things, test. You must note it up. These are the two things. Okay, confirmatory test is very, very important. The question. Okay, so next. So this is the test samples. See here how they are preparing. They are all are in the diagnostic aspects. When the person suffering from the AIDS, they went for the diagnostic test. So this is the test. Okay, enzyme immunosorbent assay. See this enzyme, how it is. This is structure already. So the mechanism we didn't discuss, isn't it? There are two types: the direct method, indirect method. It's simply complicated one. When you are going to know the mechanism of the Western blot or ELISA, okay, you go through it, you will get the ideas. Okay, so these are the things: antigen, antibody, enzyme, and also substrate. When it enzymes combine with the substrate molecule, it will produce the product. Suppose the product is in color is changed, blue is orange. So when the color change, the person suffering from the AIDS. Suppose if it is not changed, it is not a confirmation. Okay. Suppose one more is that. Suppose the color changes also depends upon the directly proposed to the severity of the diseases. That is all. This is the primary antibody, secondary antibody, capture antibody. There are so many things. Don't need these things. Okay. Types of ELISA: direct ELISA and indirect ELISA. That's why I told you sandwich ELISA and competitive ELISA. These are the four types have been given. Already in our what is the ELISA already here what they have there are four types have been reported according to their mechanisms, how it will be acted. Okay. Even so many diagnostic companies also that Dr. Reddy. Okay. So many things is there. Okay. Mode of action or AIDS virus. After getting into the body of a person to the blood, the viruses enter the macrophages. When it entered the virus into the particular person, it may first enter the macrophages. What is meant by macrophages? WBC is the macrophage, isn't it? Because any one of the pathogen enter in the body, first who will fight it? The macrophages in the particular WBC only fight on the macrophages. Macrophages fight on the particular pathogen. Okay, suppose if it is not possible, they will hand out to the which one, T lymphocytes and B lymphocytes. Suppose if it is not possible, what happens? They cannot do anything. Finally, they lead to death. Okay, that is the reason it lead to death. It means immunity power completely reduced. Once reduced means all the other diseases also enter. That condition is called a symptom. Okay, more than one disease enter into the human body or any other host, that condition is called a symptom. That is the reason. Okay, where the RNA genome of the virus is replicate to form a viral DNA, the RNA to DNA with the help of the reverse transcriptase enzyme. The process is called as reverse transcription. Okay, already we saw it. See here, this is very, very important. So many times they were asked. DNA produces the DNA molecule. Okay, that condition is called as replication. The exact copy of the DNA is produced by the DNA molecule. The condition is called as replication process. Another one is called as when the DNA molecule produces the RNA, that condition is called as transcription. The double standard DNA molecule produces the single standard RNA molecule is called as transcription. When the RNA molecule produces the double standard DNA molecule, that is called as reverse transcription. Here you just note it down, DNA to RNA. But in this case, RNA to DNA. It happens in the where? In the HIV category, pathogens present in the human body. It will be transferred the RNA to DNA molecules. And then DNA addition with the RNA molecules. What are the types of RNA molecules? There are three types of RNA molecules. One is called tRNA, RNA, and mRNA. These are the three things, isn't it? Along all these molecules, nucleic acids are united together and used for the protein synthesis. That protein synthesis is called as translation. Okay, that's why the protein synthesis, that is called RNA, is called as which one, and rRNA, tRNA, ribosomal RNA acts as a protein factory. Okay, so these are the very, very important. Just note it in same time they were asked the first thing from this NEET, DNA to DNA replication, DNA to RNA transcription, RNA to DNA reverse transcription, and DNA plus RNA. Okay, which one? The protein biosynthesis takes place, which process is called as translation. Okay, this is see that the viral DNA gets incorporated into the host cell DNA and directs the infected cell to produce a virus particle. The virus particle is nothing but the antigen. The macrophages continue to produce virus, and this they act like a HIV factory. Simultaneously, HIV enter the alpha T lymphocyte that is denoted TH, below 200 nanometer replicate and produce a progeny viruses. That's the things already we discussed. This is the main mechanism of the HIV virus. When the particles, a foreign particle enter, where it attacks. Okay. After that, what will happen now? Normal production of T lymphocytes, it will be reduced. As a result, what happens now? Your immunity power is also reduced simultaneously. Okay. See, the progeny viruses released in the blood attack other helper T lymphocytes. That's why the immunity power completely lost out. Okay. Suppose 1 hour or one or after the incubation period, take an example of 3 months also. After that, what will happen? Automatically they produce more number of viruses. The more number of virus production takes place. As a result, the more number of lymphocyte production will be reduced. Finally, what happened? Our immunity power is completely lost. That condition is called a syndrome. This is repeated, leading to the progressive decreases in the number of helper T lymphocytes in the body of the infected person. We told you. During this period, the person suffered from about of fever, diarrhea, and weight loss. So this is a simple thing. Okay, about fever, diarrhea, weight loss, sweating. Okay, suddenly they increase the temperature also. Okay, and also appetite, headache. The things also it comes. These are the symptoms. Okay, life cycle of vaccine. How it will be entered? What will be the main activity? How the enzymes are activated? All those things already discussed. Once again, repeat for you. This is also a life cycle of HIV. After getting into the body, the HIV enters the macrophages of T helper cell. The viral RNA genome replicates to form a DNA. That condition is called as reverse transcriptase. With the help of the enzyme is called as reverse transcriptase. Okay. Then the viral DNA gets incorporated into the host cell DNA by an enzyme called integrase. How it will be joined with the DNA of the viral molecules, virus molecules into the host DNA with the presence of the enzyme is called as integrase. Okay, two DNA molecules are incorporated. Okay, by the enzyme is called integrase. That is very, very important. Sometimes they were asked a question. How it will be the host DNA molecules incorporated with the viral DNA molecules with the presence of the enzyme is called as what is that? Integrase. Note it up. Okay. So it is also and also direct and infect the cells and produce the virus particles. When it incorporated, takes place automatically in our transcription reaction also possible. So as a result, it produces the exact copy of DNA molecules. Which one when it incorporated occurs, both the DNA. As a result, the viral particles also produce the viral DNA, isn't it? Again, the viral DNA controls all the activities of the host DNA and produce their own specific viral genome. As a number of viruses also multiplication takes place. Here, this is the life cycle. First of all, you should know there are how many steps here. There are 7 steps have been reported here. First one is binding of the receptors. The fusion takes place with the help of the glycoprotein. It's a hook-like structure. This is called as receptor, isn't it? Easily bind with the glycoprotein 120. Note it down, very, very important. And after that, what happened in the fusion also takes place, fused insect. So inserted means what happened? It may be introduced the RNA molecules. When it introduced the RNA molecule, it entered into the host DNA molecule. Here, with the help of the process is called as the reverse transcription. What is the meaning of reverse transcription? When it binds with the receptor, it enters inside. Finally, what happens? The single standard RNA molecules produce the DNA molecule. The DNA molecule enter inside and also integration takes place in this area. In this area, within the host nucleus cell. So it's bind to here the blue. After that, it produces a replication process. Replication process. After the replication process, assembly also takes place. This is already studied in the virus protein molecule comes out. these are all again the another the lifestyle feature what will happen they may be easily again and again repeated. Hello, ma'am. Sharing. Excuse me. Can you hear my voice?
Yes, sir.
Yes. Yes. Okay. Thank you. Any doubt in this area, ba? Do you have any doubt in this area? Okay, just follow it properly. I think today I am very too fast, isn't it? Okay. Anyway, just follow it. Suppose if you have any doubt, please ask a question. Please say to stop.
No, sir. No questions.
Okay. Okay. I understand. Okay.
Sir, are you there? Sir, you are not audible, sir.
Sir, you're not audible, sir.
Okay. இப்போ கேக்குதுங்களா? Okay, okay, thank you. Okay, okay. T help. That's why when it attacks the vessel, it will not produce the T help. Okay, there is a progressive reduction in the number of T helper cells. Due to the reduction of T helper cells, the person starts suffering from the infection of other viruses. Okay, when it reduces the production of T helper cells, what happened? The person starts suffering from the infection of other viruses. Other viruses already told you so many things is that not only that and fungi and also even a parasite like plasma. These are the things. That's why it is called a syndrome. The patient become immunodeficiency and more prone to other diseases also. So that condition is called a syndrome already we discussed. See, so this is the structures already given in your book also. The retrovirus. This is the viral RNA core. The virus infects normal cells, or this is called as normal cell. This is considered a normal cell. When it bind with what happened? The nucleic acid is also transfer inside the host cell. It is also incorporated with the host cell. Finally, what happened? Replication process with the help of the enzymes, RNA to DNA, DNA to again RNA molecules. It produces the nucleic acid. Okay. Finally, the protein also synthesis takes place, and also finally the assemble all the molecules, and it produces the new viruses, and it also comes out from the cells after the lysis of the cell wall. Okay, so this is the thing. Due to the decrease the number of the helper T lymphocytes below 200 millimeter cube, the person starts suffering from the infection. So you must know the normal value of the T lymphocytes in your blood. Okay, suppose if it is ready to 200 millimeter cube, that is called as what is that? Normal level, I think. Suppose if it is reduced from the 200 millimeter below, means what happened? You are in the sick condition. You are suffered by an HIV, any other virus like that. That could have been otherwise overcome, such as those due to a bacteria, especially mycobacterium. Already told you, HIV is a good friend of mycobacterium, otherwise called as TP. Okay, viruses, fungi, and even parasite like a Toxoplasma. These are all very, very identified components in the HIV process. Okay. Go to the next one. Third phases, asymptomatic phases. Sometimes the symptom is not overcome out. 2 to 10 years, there is no antibody production in the first 2 to 12 weeks. So infectivity of the phases or activeness of viruses maximum in this period. 2 to 10 years, there is no symptom. That's why that condition is called as asymptomatic phases. This period is called a window period. That's noted down. And 95%, not only that, 75% of the people, they will not go for the, asymptomatic nature only. So this period is called as window period. Sometimes they may ask the question, window period, how many weeks or how many years? Like that, they may ask the question. Okay. No specific symptom appear in the patient, so the ELISA test is negative. Once again, repeat for HIV person. Okay. ELISA test is negative that condition. When the windows period. Okay, note it on this one. So the window period is how many days, how many weeks, years. Like that, they may ask the question, which period, what happened? The ELISA test will be negative, like that they may ask the question. You just understand this is the thing. And second one is, and B is the second category, the AIDS related complex, ARC, 3 to 6 weeks. Diarrhea is possible, weight loss is 10%, cough, and generalized lymphadenopathy, and also fever. These are the second week of related AIDS related to complex. See, and then a full-blown AIDS. Person completely occupied by the HIV molecules, completely suffered by the HIV molecules. How many percent is 100% means what will happen? What will be the symptom of the person? Patient become fully immune deficient in this period. This is the first symptom. So what happened? Immunity period reduced means what happened? The T lymphocyte, these lymphocytes production will be reduced in nature, isn't it? Okay. How it will be? Already we discussed the mechanism of the HIV active and the cells, particularly T lymphocyte. T lymphocyte, there is a receptor, is the CD4 count is also reduced. How much? See here, this is the less than 200, okay, and also 10 power 6 liter mycobacteria. Okay, so this is the number. When it reduces the normal level, that is called as what is the symptom of second clinical symptom. And third one is called as Mycobacterium avium, pneumocystis cami, pneumonia, lungs infection, toxoplasmic encephalitis. Ancelity will discuss it. Toxoplasmic encephalitis. It's nothing but the memory loss. Okay. Kaposi sarcoma. Kaposi sarcoma is also another type of AIDS virus. It became an oncogenic virus. It's similar to that of the oncogenic virus. It is a, which one you know? Cancer producing, like it causes cancer or blood vessel. So it may be lead to the lymphoma. What is my lymphoma? Already know about. Otherwise called as leukemia. What is my leukemia? Nothing but the blood cancer. Okay. So skin appear purple in this cancer. These are all are considered getting full blown AIDS. Okay. So once again repeat for you. Patient become fully immune deficiency in this period. T lymphocytes also CD4 count will be reduced less than 200 and 10 to the power of 6 liter mycobacterium. Other diseases also occupied. What are the mycobacterium? Otherwise called as TP, pneumonia, particularly lungs diseases. Toxoplasmic encephalitis is nothing but the memory losses, and also the water also filled with where the brain cells. Kaposi sarcoma is another type of AIDS viruses caused by the affected person. Okay, it causes cancer or blood vessels. Skin appear what is that? Purple in this cancer. Okay, see that treatment. This is very, very important. There is no treatment, not at all identified. Okay, nowadays the research is going on. The monoclonal antibody, like that, it produces a single type of antibody production is also going on. Okay, so listen here. The treatment for AIDS, the treatment of AIDS with the antiretroviral drugs only. The only particles partially affect because it can only prolong the life only. Okay, there is no permanent cure. The person but cannot prevent death. Okay, those who are suffering from the AIDS, the person completely, what happened once he will lead to death only. They will not curable. Okay, so it may be extended their lifespan only. That is the reason. How it will be? There are two things have been reported. Reverse transcriptase inhibitor. Here, what is by reverse transcriptase inhibitor? Already we discussed so many times. Will you please anybody else? What is by reverse transcriptase inhibitor? Reverse transcriptase means already know. RNA to DNA, isn't it? By the enzyme is called as reverse transcriptase enzyme. Suppose the reverse transcriptase enzyme inhibited by some of the component, what happened? The RNA to DNA conversion is not possible, isn't it? As a result, what happened? The protein, the viral DNA, it will not enter with the incorporate with the host DNA. That process also suffers. So as a result, new virus DNA molecules, new virus also is not possible. Production is not possible. That is the reason. So they were used this type of inhibitors. What is that? Zidovudine, previously called as acetothymidine. So many times they were asked the question. AZT is the expansion. What is that? Which one of the following is the expansion of AZT? And previously, what they called? Okay, this is the acetothymidine. It can expand the life only. It is not curable. How it will be act as it is also act as a reverse transcriptase inhibitor. Note it down. This is very, very important. And another one is called as DDT. Not here. Didexamidine. Didexamidine. Force current drugs inhibit the enzyme reverse transcriptase of HIV, like this. Okay. So both are activities also similar, but the component will be varied. Chemical composition will be varied in nature. This is also a very, very important. And then protease inhibitors. Another one is, suppose the hooks, it is not bind with the CD4 receptor. It can be prevented by some of the chemical composition. Suppose if it is not bind with the receptor cell, means what happened now? It will not enter the genetic material inside the host cell. So as a result, what happened now? It can be prevented. It can be prevented by the production of the new genes. That is also another important thing. Okay. See here. Protease inhibitors, highly active antiretroviral therapy, included both reverse transcriptase inhibitor and also protein inhibitor drugs. So there are 3 drugs have been reported. One is called as acetothiamidine, otherwise called as zidovudine. Second one is called as DDT. Okay, what is the main mechanism of this one? We completely inhibit the production of which one, reverse transcriptase enzyme only. Okay, and third one is called as protein inhibitors. Okay, these are the three drugs they were used for the HIV approach. How will you prevent HIV? You know very about that. AIDS has no cure prevention, the best option. AIDS, we will not completely curable because it completely, once it enters, the person completely lead to death only, until the death only. Okay. National AIDS Control Organization, they organize so many programs, NGOs, educated the people how the AIDS is coming. Okay, how it will be like that. NACO, established in the year 1991, and other non-governmental organizations, that is called NGOs, educate the people about the AIDS. Awareness of the AIDS program also conducted by these organizations. And also, save blood for transfusion. This is also very, very important. So many blood bank is also there. They also transferring the blood, isn't it? So you have to test whether the blood is whether it's a good one or bad one, like that. Use of disposable needles. Suppose if nowadays there are so many hospitals, they were used only one time one day. That is called disposable, isn't it? Suppose any person suffering from the AIDS, that same blood, same needle is also used for the another person. What will happen? It will be easily entered. Like microorganism, it's very dangerous. Particularly HIV is a very one. That's I use the disposable needles, and also here the blade. Okay, and take an example of another things also. Prevention of drug abuse. Prevention of the drug abuse. Don't use the drugs. Advocating the safe sex and promoting the regular checkup also. These are all the prevention of AIDS. Okay. So once again, you go through it, you will get it the exam. So next class we will see the remaining on the which one cancer and the science, the study of the cancer is called oncology. We'll see that thing. See today what we have discussed. One thing is to remember what is by AIDS, acute immunodeficiency syndrome. What is the cosmetic organism? What is that? HIV, human immunoviruses. What are the main structure of the HIV? Okay, who discovered first? How many people are affected? And then what are the structures already in our report? See here, this is called as retrovirus already in our report. It has two type of protein molecules. The upper hook-like structure is called as glycoprotein 120, and below there is a needle-like structure is called as glycoprotein 41. Inner side, that bilipid layer is also present in it, and then the capsid is also present. It is called as matrix. Inside there is a single stranded RNA molecule with the presence of the reverse transcription enzyme. Not only that, and also the another enzyme is called as integrase enzyme also. The integrase enzyme only helps for the incorporation of DNA molecules of the viral DNA as well as and which one post DNA. Okay, already know about that things also already. What is the main who discovered already? What is the related viruses or lymphoadenopathy associated virus, and also human T lymph cell lymph. Okay, and leukemia viruses also. These are all the related to the retrovirus category. Why it is called as retrovirus? Already I have told. What is the structure? What it will happen? What will be the internal part? Or what will be the outer part also? Already we discussed, isn't it? Okay. And the next one is what is that? What will be the structure? Structure of protein already we discussed. What are the protein molecules? What are the enzymes? Okay. And then machinery aspects. What will be the mechanism? How it will be entered into the human body? Already we discussed. So it can be and also transmission. Transmission is very, very important. These are the mode of transmission. How it will be entered into the human body. Okay, like that. Sexual and also a parental blood transmission and non-sterile tools also. That is also we discussed. And also how many percentage it will be entered. Okay, it's also discussed. Okay, and then so investigation. What are the investigations already we discussed? There are three types of investigations have been reported. One is called as ELISA, another enzyme linked immunosorbent acid, it is called a screening test, and second one is called as what is that? Confirmatory test for AIDS. Which one Western blot is the confirmatory test. And as antibody test, antigen antibody test, and also nucleic acid test is also the direct method, indirect method, and also one directional, two directional, and also oral quick method already we discussed. These are the tests present in it. Mostly these are the two only that they have been used for the identification. That is also already discussed, isn't it? Okay. And then how will you prevent the mechanism already know how it will be entered, and also how it will produce the DNA to RNA, RNA to DNA, like the DNA to DNA replication, DNA to RNA transcription. Here, what happens? HIV reverse transcription takes place between RNA to DNA. That is also the thing we discussed. We were discussed, isn't it? Okay. And then life cycle already we discussed that things also here. So one is stages order, 7 stages order. One is called as budding, second one is fusion, third one is reverse transcription takes place, and then integration, replication, assembly, and that comes out. Okay. Again, the cycle will be continually going on, isn't it? That is also already discussed. Okay. And then what are the phases of the? There are 3 phases already we discussed. One is called as asymptomatic. Asymptomatic pattern means what do you have? 90% of people suffered by this symptoms only. There is no symptoms, but at the same time, the person suffered by the HIV. Okay, so they may be confirmed. They may be screen test. When they are going to the screen test, they will be identified the person is suffered by the what is the negative test. Negative result only they will get it. This is also another thing. And also, and 3 to 6 weeks after the receiving of HIV, diarrhea, weight loss, and cough, and generalized lymphoadenopathy fever also is there, isn't it? And the last one, fully bounced in what are the symptoms? Okay, this is very, very important. How it will be defined? The count will be reduced below the that of the 200. Okay, so this is also. So that's all about today. So what is the main treatment already in oral? There are 2 things that reverse transcription are both are. One is called as acetothymide, another one is called as DDT. This is very, very important. Last one is called as a protein, isn't it? So that's all about today classes. We'll see the remaining things on the next class. Thank you. Bye. Take care.
Thank you,
Any
sir.
doubt in this area? Okay, okay. Bye.
No
Take
doubt,
care.
sir. Thank you,
Okay. 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 Bye.